Welcome to another the eBiology Teacher Podcast. I'm Mr. Doc, and today we are talking about mitosis. Let me go ahead and get all the way up to the front here so we can go over what it is that we hope to learn today. All right. Now, one of the things we're going to do is talk about how chromosomes are made up. We're going to differentiate between the term haploid and diploid, which are terms you're going to hear a lot of in the coming weeks. We're going to talk about the phases of a life cycle. We're going to talk about why mitosis is important, and we're going to talk about the phases of mitosis and what's happening at each phase. One quick important note is that all cells have the ability, or all organisms have the ability to grow. That doesn't necessarily mean that their cells are just getting bigger. It usually means that their cells are increasing in number, which means that we have to have the ability to produce new cells. Now, anybody who's ever been cut or injured knows that your body can heal itself. What happens is we damage some cells and those cells are replaced by new cells. So, that's what we're really going to be dealing with today, is understanding how that process really works. All right, so let's first talk about chromosomes. Chromosomes are often associated with DNA, but not a lot of people know exactly what a chromosome is. A chromosome is a rod-shaped structure that's made up of DNA and proteins that contains all the genetic information for a cell, which means that chromosomes are responsible for everything that happens in a cell. They have a very complex structure, and there are three reasons why. Number one, they do have to make it easy to reproduce. Number two, they have to be protected, because if all of the information for a cell is stored on the chromosomes, if the chromosomes get damaged, you are in a bit of trouble. And finally, there's a lot of it. So we have to be able to condense it down into a very small bit of area. So that's what we're talking about in terms of the structure of a chromosome. It does have to be very safe. It has to be easy to replicate, and it has to be tightly wound so that all that information can be sort of condensed. Now, the structure is pretty simple, except that it's compound, which means that it's redundant. It coils upon itself. So, the first thing we think about is the double helix, which is a coil in of itself. But what we do is then take that and wrap it around something else. Now, let me give you an example. Here is my phone cord. Imagine that this is DNA. Now, that's a lot of DNA, right? But to make this smaller, all I would have to do is wrap it around something, and then all of a sudden it's much more manageable because it's a lot shorter. Now, what happens is that my finger here is acting as a histone. A histone is a protein that allows us to spool DNA around it, kind of like a spool and thread. But then what's going to happen is this structure is going to wrap around other structures so that the more coils we get, the more room we're saving. Now, those large rod-like structures are called chromatids. We take two identical chromatids, we smack them together with what's called a centromere, and we end up with a chromosome. So if you look at this picture over here, the one with the orange background, this is what we generally think of when we think of a chromosome. That's what they look like. There are two chromatids here. One is here, and the other is here. The centromere is located right here in the center, and they're kind of stuck together. Now, it's important to note that the chromosomes are identical, which means that this guy over here and this guy over here have the same information. That's going to become important later. Now, when it comes to chromosomes, humans have 23 chromosomes, but here's the thing. Our body cells, or somatic cells, have actually 46 in total. Why? Well, because if we're going to reproduce, that means I'm going to have to share my genetic information. If I'm going to share my genetic information, then what's going to happen is I'm going to have to give it away. If I give away all my genetic information, then I don't have any for myself, and then I'm in a bit of trouble. But if I have two copies, I can give one away and keep one for myself. All right, and that's what's called diploid. Our body cells are diploid, and we refer to them as somatic, S-O-M-A-T-I-C, somatic cells. Other cells which contain only one set of chromosomes are called haploid, and these are generally referred to as gametes, the best example being that of a sperm or an egg cell. So the N number, or chromosome number, is pretty simple to understand. It's the number of chromosomes. Our N number is 23. So most body cells have 2N, which is 2 times 23, 46. But our haploid cells, including our sperm and egg cells, are haploid. So diploid cells are 2N, haploid cells are 1N. 1 times 23 is 23. So let me say that again because I think I messed up. I think I said haploid, 1 meant diploid. Diploid cells have 2N. Our chromosome number, or N, is 23. Diploid cells, which are somatic cells of the body and the cells, 2N. 2 times 23 is 46. Haploid cells, like sperm and egg, are 1N. 1 times 23 is 23. All right? And again, the reason for that is the diploid cells need to be able to give away 1 and keep 1. Now, the cell cycle is a series of repeating events that happen in the life of a cell. The majority of a cell's life is spent in interphase. Interphase being that time where it's just been formed, the cell's just been formed, till the time where it's ready to produce. And so right here on our little diagram, we can see here that there are 3 subphases to interphase. The first one is called G1. This is the growth phase. This is where the cell is starting to reproduce those organelles that it needs and all that kind of stuff. In the S phase, we are left with the DNA copying, and we've already discussed that when we first started talking about DNA replication. S phase is where the DNA is going to copy, and we're going to make sure that we have enough to hand out to the next cell. And then finally, G2 is our growth and preparation phase, which is when the cell's really starting to gear up and get ready for cellular division. In cellular division, there are 2 categories. One is mitosis, and the other is cytokinesis. It is mitosis that we're going to spend a particular amount of time on today. But also cytokinesis is important because cytokinesis is where cellular division ends. At cytokinesis, we have gone from 1 cell to 2 completely separate cells, at which time interphase starts all over again. So cytokinesis is the end of the cell cycle, which starts a new round of interphase. Now, mitosis is simply cellular division, but more specifically, it's the process by which a cell divides all of its nuclear content, which would be its DNA, and its cytoplasm and organelles into 2 identical cells. This is a form of asexual reproduction. If we put the letter A before something, it usually means not or without. So something that's atypical is not typical. Something that is asexual is not sexual or without sexual reproduction. Meiosis, or meiosis, depending on how you pronounce it, is how we do haploid cells. But mitosis results in finally diploid cells. So meiosis will result in cellular division in terms of having a haploid offspring, whereas mitosis will end up with 2 genetically identical cells that are diploid in nature. Mitosis occurs only in eukaryotic cells, which are everything that we know of except for bacteria. Bacterial cells, or prokaryotes, undergo a similar process called binary fusion. Now, we're not going to get it, or fission, excuse me. We're not going to get it deep into binary fission, but you do need to know that the process of which bacteria replicates and the process by which all other things replicate are different. And binary fission happens much faster, which explains why bacteria can grow much quicker than, say, some other cells like ours. Okay? Now, the phases of mitosis are pretty simple. There are 4 of them, and I remember them this way. P-MAT. P-M-A-T. The 4 phases of mitosis are prophase, metaphase, anaphase, and telophase. Prophase, metaphase, anaphase, telophase. P-MAT. Now, I would figure out a way to remember that because this is a process. And for metaphase to happen, prophase had to have happened. So we can't skip a step. So if we get out of order, something's not going to happen correctly. So come up with a mnemonic device. Somebody in class, and I probably shouldn't say this, but I'm going to anyway, came up with a mnemonic device. Pimps might act tough and crazy. P-M-A-T and C. I thought that was creative. All right? Anyway, let's talk about the phases. First of all, prophase in mitosis is the first step. In prophase, the word that I associate with prophase is packing. If you're going to go on a trip, you're going to pack. In the nucleus, we're going to have all the DNA hanging out, And then when it's time to undergo prophase, everything's going to get packed together nice and tight. So if we were looking at a cell under a microscope that was in interphase, we wouldn't see these chromosomes. But in prophase, the chromosomes start to condense down and they do become visible. Also what happens is the centrioles start to go to opposite ends of the cell, and we start to create or start to build spindle fibers. These are the fibers that are going to give the cell its structure during mitosis. During metaphase, which is the second phase of mitosis, the nuclear membrane dissolves and the chromosomes start to line up at the center of the cell. So if prophase is packing, metaphase is middle. The nuclear membrane disintegrates, everything starts to line up at the center. Those spindle fibers that started creating, that have been started to build during prophase, are going to be connecting, forming what's called the mitotic spindle. This is kind of the skeleton for cellular reproduction in terms of mitosis. The third phase, and by the way, P, prophase, equals packing. M, metaphase, equals middle. A, anaphase, equals apart. The third phase of mitosis is anaphase, in which those chromosomes break apart at the centromere, and one chromatid from each side goes one to each side. So we're talking, they separate. And they're going to the poles of each other cell. Poles as in north and south pole, not poles as in flagpole. Okay, so we're going along. And then what's going to happen is as they move apart, the cell is going to elongate. It's going to get longer. Okay, and then what's going to happen is each cell has one set of chromatids. Finally, we have the telophase. Ta-da! That's the only T that I could come up with for telophase. The fourth phase of mitosis is telophase, and in this, chromosomes reach the opposite poles and begin to recoil. This means that the nuclear membrane is going to form, and we're going to start to look like two separate cells. At the end of telophase, cytokinesis happens. And here's what's happening here. This little bit right here is called the cleavage furrow. And what's happening is we have this elongated cell, and it's starting to pinch in on itself. And then at the end of cytokinesis, boop, we have two separate cells. So the cleavage furrow cuts the cell into half. They separate into two identical cells. That is prophase, packing, metaphase, middle, anaphase, apart, telophase, ta-da, cytokinesis, cut. And there you go, two separate cells. Now, let's list the components of chromosomes from smallest to largest. First of all, we start with DNA. DNA coils around histone proteins to form chromatids. Chromatids attach together at a centromere to form a chromosome. The difference between haploid cells and diploid cells are the number of chromosomes. Remember that diploid cells are 2N, which means they have an extra copy of their genetic material, so they can pass one copy off to the new cell and keep one for themselves. Haploid cells only have one set of chromosomes, and those consist of sperm and egg cells, which are called gametes, whereas diploid cells are somatic cells or body cells. The phases of the cell cycle include interphase, and interphase is broken down into three parts: the G1 phase, which is where a cell starts to grow and develop; S phase, which is where the DNA copies; and then S2, which is where the G2 phase, which is where the cell prepares for division. And then we have mitosis, which is where the cell divides, and cytokinesis, where the cellular division finishes. At the end of cytokinesis, we go back into interphase, and we repeat. Why is mitosis important? Well, if anything wants to grow, it's got to be able to produce new cells. And we know from the cell theory that all cells come from pre-existing cells. So mitosis tells us how that happens. And finally, what are the phases of mitosis, and describe what's happening at each. I'm going to do you one better. If you look right here in the corner, this is a diagram. We're in prophase right now. Now we're in metaphase. Things start to line up in the middle. Now we're in anaphase. They're moving apart. Now we're in telophase, and boom, cytokinesis. Prophase starts to dissolve the membrane. Everything starts to line up in the middle of metaphase. Then what happens is anaphase, moving apart, telophase, new membrane, boom, there we go. So as you can see, that's moving, and that's of course on my website. If you want to watch this little animation here, you can watch it for days and days and days and days. You know, it's just so much fun to watch. Anyway, I hope this has helped you. Our next topic is going to be meiosis or meiosis, whichever you prefer, in which we're going to be talking about haploid cells. But the key word of the day is mitosis and diploid. Remember that, and then we'll move on from there. I hope it's helped. I'll see you in class.